Microbiological culture is the most important diagnostic technique used in medical microbiology. This is inducing the reproduction of microorganisms, these may be bacteria or fungi, on an artificial media in the laboratory. Microorganisms will multiply on this medium until they give rise to a visible colony. Each colony is derived from a single bacterium, a single fungus, or a clump of these. These colonies can then be tested in the laboratory in different ways. So, a sample from a patient is placed onto, the term is inoculated, onto culture media to determine if any organisms are present, and if they are present, what they are and how many there are. The two main types of media used are solid media and liquid culture. Solid media is made in a petri dish like this and contains a solidifying agent called agar. Only solid media will allow the production of single colonies of microorganisms, and so is the more important of the two. Liquid or broth culture is used less, but does have some applications which I'll discuss later. There are hundreds of different types of media available, and the reason for this is that different species of bacteria and fungi will vary greatly in their nutritional requirements. And this is linked to the huge variety of metabolic pathways utilized by different species of microorganism. So we need to match the nutritional requirements of any bacteria or fungi we are interested in and provide these in the media. Now, some microorganisms can utilize a large range of nutrients and will grow very easily in the laboratory, while others have more exacting nutritional requirements and are more difficult to grow. These are known as fastidious microorganisms. The simplest media contains a source of carbon, such as glucose, plus something like beef or yeast extract to provide nitrogen and amino acids, along with trace amounts of various salts. An example of a simple or basal medium is nutrient agar or tryptone soya agar, as shown here. Non-fastidious organisms such as Staphylococcus aureus and Escherichia coli will grow easily on this medium. Enriched media contains a basal media which is enriched with a substance to provide extra nutritional factors which allow the growth of more fastidious microorganisms. So, for example, blood agar also contains 5% sheep blood, and the nutrients and growth factors found in sheep blood help to support the growth of, for example, Streptococcus pyogenes and Streptococcus pneumoniae. Another enriched medium is chocolate agar, which does not actually contain chocolate, but instead is another type of blood agar. In this case, the plate has been heated to allow the red blood cells to lyse and release their contents. This medium is useful for the growth of, among others, Haemophilus influenzae. It is important to know where in the body the samples come from. This is not only because some pathogens only cause infection in certain sites of the body, but also because some parts of the body contain microorganisms even in healthy people. So, for example, the gastrointestinal tract contains billions and billions of bacteria. Other sites in the body which contain microorganisms in health are the skin, the upper respiratory tract, including the nose and throat, and the female genital tract. So this poses a challenge for microbiologists. How do you find a pathogenic microorganism among the billions of other microorganisms present? It's like looking for a needle in a haystack. And the answer is to use selective media. Selective media contains something which prevents or inhibits the growth of many of the microorganisms which we are not looking for, while allowing the growth of the microorganism we are looking for. An example of this is Campylobacter media. Campylobacter is a genus of bacteria which causes gastrointestinal infections. This media is made selective as it contains a cocktail of antibiotics. Of course, the choice of antibiotics is important. The microorganism we are looking for needs to be resistant to the antibiotics used so that it will grow on the medium, while other microorganisms should be sensitive and therefore would not grow. Now this is Sabouraud's agar, and this is a selective medium for the isolation of fungi such as dermatophytes, which cause skin infections, and Candida albicans, which causes a number of different infections. In this case, it is made selective as the pH of the medium is acidic, around 5.6, and this will inhibit the growth of many bacteria. Another selective medium is MacConkey, which is used in the isolation of gastrointestinal pathogens such as Salmonella, and those which are often found in the gastrointestinal tract such as E. coli. This contains bile salts, which inhibit the growth of many bacteria. However, MacConkey has another feature. It's not only selective, it is differential, which means it can be used to differentiate between similar bacteria which have different metabolic properties. And in this case, it will differentiate between bacterial species who ferment the sugar lactose with those which do not. And so, MacConkey is known as a selective differential medium. Chromogenic media is a relatively recent development which is becoming widely used in laboratories. These types of media depend on us knowing a specific metabolic characteristic of the microorganism of interest. How this works is that the multiple modified substrates are incorporated into the media. The substrates are modified by the addition of a chromogen, a molecule which produces colour when it's cleaved. So each substrate is matched up with a particular microorganism of interest. If that microorganism is present, it will utilise the modified substrate as it grows. Therefore, the modified substrate will be hydrolyzed, releasing the chromogen and producing a coloured colony. And so the specific colour of the colony is an indication as to the identity of the microorganism. Robertson's cooked meat is an example of a liquid culture system which is used in the laboratory. It is exactly as it says, it's sterilised cooked meat and is used to allow the growth of fastidious microorganisms. The idea here is that a patient sample is inoculated into the cooked meat and this is incubated to allow any bacteria to grow. After 24 hours growth, a portion of this is transferred, the word is subcultured, onto solid media such as blood agar. After 24 hours in cooked meat, bacteria present in the sample have had the opportunity to multiply, increasing their numbers, and so there is a better chance of recovering them on solid media. So this technique is important as it is improving the sensitivity of culture. This is especially useful for the isolation of anaerobic bacteria as they may become injured due to exposure to air when the sample was collected. Another liquid medium is alkaline peptone water, which is used to improve the recovery of Vibrio cholera, which causes cholera, a disease of the gastrointestinal tract. This medium is alkaline, as Vibrio cholera grows well under alkaline conditions, whereas most gastrointestinal bacteria do not. So this process helps to enrich for the presence of this species. So we've covered the basic types of culture media and provided examples of how they're used in the diagnosis of infection. Now, what also needs to be considered is the temperature and atmospheric conditions under which these plates are then incubated. So many organisms such as E. coli, Staphylococcus aureus will grow very happily in atmospheric air at 37 degrees C. For others, we need to provide a special atmosphere. Many fastidious bacteria prefer a carbon dioxide enriched environment. So, for example, if we suspect Streptococcus pneumoniae is present, we will incubate in an incubator containing 6% carbon dioxide. Some bacteria, known as anaerobes, are killed in the presence of oxygen, and we need to incubate these in an oxygen-free environment. An example of a pathogenic anaerobic bacterium is Clostridium difficile. Finally, while most pathogenic bacteria grow optimally at 37 degrees C, fungi prefer to grow at a lower temperature. And so when we attempt to isolate fungi, typically we would use an incubator at around 28 degrees C.